Hi, well, welcome everybody to the November meeting of the Ohio State Emeritus Academy. Before our talk today, I want to have just a very brief business meeting. And the first thing is that I will formally introduce new members to the Emeritus Academy next month. But I want to acknowledge that one new member, Hans Klompen from EEOB, is on this at the meeting today. I want you to, if you could please, we did not have very many people apply for membership in this cycle. And so if you know of any of your friends or colleagues who are retired and would enjoy being part of this group, please encourage them to apply in the next cycle. The next steering committee meeting will evaluate small grant applications that were due on Monday. And I also say that our next meeting will be on Wednesday, December 1st. And our speaker will be Marshall Unger from the Department of East Asian Languages and Literature, whose presentation will be Misunderstanding Chinese Characters, What's Been Learned Since 1998. We also have a November newsletter coming up. The deadline for this is November 22nd. And please consider submitting a short narrative with a photo of your ongoing scholarship, any awards you've received recently, etc. And even though the deadline is not until November 22nd, I'm sure that our dean would welcome all early submissions as she tries to assemble this newsletter. So during our talk today, please mute yourself. And then after the talk, I will attempt to monitor questions that can be asked either in person or in the chat feature of Zoom. Now I'm pleased to introduce Jackie Wood, who is an Academy Professor of Physiology and Cell Biology. Jackie received his PhD from the Department of Physiology and Biophysics at the University of Illinois. And he has had positions at the University of Kansas School of Medicine and Medical Center and the University of Nevada School of Medicine before coming to Ohio State in 1985, where he served as professor and chair of the Department of Physiology and a professor of internal medicine at the Ohio State College of Medicine. Dr. Wood has published more than 400 journal articles, abstracts, books, and book chapters during his career. And the recent focus of research has been on motility, enteric neuropathobiology, emerging concepts in neurogastroenterology, and enteric nervous control of digestive functions. His work has largely been funded by the National Institute of Health and from industry. An example of his work, here are some titles of some recent books that he has written or co-edited. In 1989, his Handbook of Physiology, the Gastrointestinal System. 1990, Frontiers in Smooth Muscle Research. 2011, Enteric Nervous System, the Brain and the Gut. And 2012, Physiology of the Gastrointestinal Tract, which is now in its 5th edition. He's received multiple awards and honors that recognize his significant research program, and I'll just mention a few. These are the Fellow of the American Gastroenterological Association, an Alexander von Humboldt Fellowship to work in Germany, and he's an honorary citizen in the city of Atsugi in Japan. He's also received teaching awards from all 3 of his academic postings: the Chancellor's Excellence in Teaching Award at the University of Kansas, the Professor of the Year Award at the University of Nevada, and the Ohio State College of Medicine Excellence in Teaching Award. And this is just a subset of the variety of honors and awards that Dr. Wood has received. And also, Dr. Wood has been extremely active in a variety of professional service roles. So it is my pleasure to introduce Jackie Wood, whose talk today is titled Research on Ulcerative Colitis, Crohn's, and Colon Cancer. Jackie, you have the floor.
Well, thank you, Ardeen. I'm happy for the opportunity to to talk about a project that I've had going almost since I arrived at Ohio State in 1985. If we could have the next slide, Jason. One of my best friends and close colleagues is Dr. Owen Peck. Owen is now, was professor of medicine, attending physician at the Washoe Regional Medical Center at the University of Nevada, Reno. Before, well, Owen is a very good basketball player and was close to being All-American at the University of Kansas when he was a student there. This picture of Owen was taken when he invited me to come along when Bill Self, the coach of University of Kansas basketball team, had a reception for some of the older players, and I went along to Lawrence. So Owen was a Kansas Buckeye. Before he was a Kansas, before he was professor of medicine at the University of Nevada, he was also a attending physician at the Palo Alto Medical Clinic in Palo Alto, California. This becomes significant because, next slide, please, because the large number of nuclear physicists that were living in Southern California in the 1940s at the time of most of the research on atomic and hydrogen bombs. Owen, when he would come, once I moved to Nevada, he would come around and say, "Jack, what do you know about ulcerative colitis?" And I would say, "Owen, I don't know much, but if we're to study something like ulcerative colitis, you'll need an animal model." Owen's interest was in some of the, a couple here shown, nuclear physicists that that worked at University of California, Berkeley during the war. Ernest Lawrence on the right is a Nobel laureate in physics. The significance for Ernest Lawrence is he had ulcerative colitis and suffered from ulcerative colitis and was treated at the Palo Alto Clinic. Also in that group was Edward Teller, also treated at the Palo Alto Medical Clinic. Teller, although called the father of the hydrogen bomb, he didn't like that at all and was very much anti-nuclear bomb development following Hiroshima and Nagasaki. Edward Teller was at a conference, Owen told me this story, but at a conference in Geneva, Switzerland, campaign against atomic war. And when at there, the stress of at the conference and presenting his opposition to the hydrogen bomb, although he was referred to often as the father of the hydrogen bomb, development, exacerbation of his ulcerative colitis. They brought him back to Palo Alto. He developed a toxic megacolon and passed away in 2003. Next slide, please, Jason. Well, I mentioned to Owen that we should have an animal model in order to effectively study a disease such as inflammatory bowel disease and ulcerative colitis. I was at a study section meeting in Washington a long time ago, shortly after I'd come to Ohio State, and I heard and was told about a new model for colitis, for spontaneously occurring colitis, and as well as colon cancer in a small primate model called the cotton-top tamarin. The cotton-top tamarin, there was a colony at Oak Ridge, Tennessee, and this was where the colitis and colon cancer was first reported. There was a colony of a little over 30 monkeys. I told Owen and we decided we had to have that colony. We acquired it through grateful donations from inflammatory bowel disease patients and moved the colony to Ohio State after I was in residence at Ohio State. Next slide, please. On the right is a endoscopic, I'm sorry, on the left is an endoscopic view of the lumen of a normal cotton top tamarin's colon. On the right is an endoscopic view of a cotton top tamarin's colon, but it has ulcerative colitis, spontaneously occurring colitis. For the normal on the bottom is the histology, which are normal histology, mucus cells filled with mucus, an inflammatory infiltrate that's not excessive. Whereas in the colitis, biopsy from the colitis colon, there is, first of all, a depletion of mucus, none of this red stuff that's here, dense inflammatory infiltrate, and mucus and mucosal abscesses. Next one, please. The other significant finding at the Oak Ridge University for the cotton top tamarin was the cotton top tamarin with colitis also develops colon cancer associated with the colitis. On the left is an X-ray of the blocked colon of a tamarin with a tumor, and on the right is the autopsy from that animal showing the opened colon and the tumor within the colon. Next slide, please. Well, most of the work that I'll be telling you about in Colombia and at Ohio State involved essentially endoscopy to access the colon and biopsy of the colonic mucosa. Here's Dr. Peck again demonstrating that the endoscopic procedure to our Colombian host and a plantation worker at the Monterey Forestal. Also, it's in Colombia. And I mention this because it was the Monterey Forestal company that thought it was good to have something positive for Colombia at the time of Pablo Escobar and the drug cartels. So the Monterey Forestal hosted us during most of our research in Colombia on wild living cotton top tamarins. The next slide. So with the colony, once at Ohio State University, we did endoscopic observations and collected biopsies. And Professor Harry Sharma in the Department of Pathology at Ohio State did the evaluation of all of our biopsies. Here is the return. Here's data for 37 of the tamarins that we had at Ohio State University that we'd had in captivity for about 1 year after acquiring the colony. And the significance here is that severe colitis and moderate colitis occurred in over 90% of the animals in the colony at Ohio State, with no mild or normal tamarins, just the severe colitis about in over 70% and 23% of moderate colitis. So something was happening in the Ohio State colony that resulted in ulcerative colitis. The next slide, please. I can't see the date there, in I think about 2000, we published a paper titled Evidence that Colitis is Initiated by Environmental Stress and Sustained by Fecal Factors in the cotton top tamarin. The next slide, please. The natural habitat for the cotton top tamarin is shown here on the right, is in upper northeastern region of Colombia, South America, and a small area of Panama. It was in this region that we had decided that we needed to investigate wild living cotton top tamarins and compare the incidence of ulcerative colitis or inflammatory bowel disease in the wild living tamarins. The next one, please. Most of this support we received for this work, I mentioned already, the Monterey Forestry Company. The Monterey, there's tree plantations for lumber for plywood is one of their products. And the second was skins from crocodiles and caiman reptiles. With this kind of activity ongoing there, they had laboratories because they breeded the crocodiles and cared for them, their nutrition, etc. So we had a laboratory area and a place to do our work that was provided by the Monterey Forest Hall. They also provided lodging for us on site and food and that sort of thing. The support listed at the top is Indarina. This is the National Research Institute for Renewable Natural Resources and the Environment in Colombia, South America. It was required for us to receive permission from them for the project, and that was not straightforward, I can assure you. They would be the equivalent of the Fish and Wildlife Service in the United States. Also, I mentioned the Monterey is significant support for us for many, many trips to Colombia for this project. They would pick us up at the airport, and if security was tight because of some of the political problems in Colombia at the time, had small airplane would fly us to the Monterey plantation. Also, I have to mention that the work was supported by the National Institute of Health by an R01 grant. Next one, please. The way in Colombia, we captured the animals were obtained by live capture, live animal traps baited with fruits from the tamarins, and we had a crew then that manned the traps continuously, hidden in the brush, and would then transport captured tamarins to research site. Usually within 1 to 2 days, we had done the endoscopic observations and collected biopsies, and the tamarins were then returned to the wild by our crew. The next one, please. This is our main team. In fact, on the left is Vicente Rodriguez. Vicente was an employee of the World Wildlife Association. There, you know who. This is Consuelo Rodriguez. She is an oncologist at the National University in Bogota, in Colombia. Miguel Rodriguez, one of the managers at the Monterey Forest Hall. Again, my colleague, Owen. Jorge Hernandez here was a world-renowned expert on small world primates. And Caballo, the horse, as we called him, was in charge of our crew that manned the traps, collected the tamarins, and got them into the lab, and then released them back into the wild. The next slide, please. So here are data from wild living cotton-top tamarins for 88 tamarins. And the data are in terms of the numbers of biopsies that were collected from each of those 88 tamarins. And here's the data for captivity at Ohio State for our 37 tamarins that we maintained here. Notice that in the wild living cotton-top tamarins, I was very much concerned that there would be some infectious colitis and inflammation in the wild living tamarins that we wouldn't be able to distinguish from spontaneously occurring inflammatory bowel disease or ulcerative colitis. But it turned out we never found severe colitis in the wild living population. And here are the data compared to Ohio State, where over 90% of the animals in our colony had severe ulcerative colitis. The next slide, please. Again, I pointed out where the natural habitat for the cotton-top tamarin is in green in the right slide. The natural habitat, this overlaps, and it was a very interesting time in Colombia when we were doing for 10 or 12 years of doing this work, because this was also the territory of Pablo Escobar, the notorious notorious drug lord in Colombia. We never encountered Pablo Escobar, but we did visit his ranch once and were just impressed with what was one thing that was interesting about is that he had an enormous ranch with small railroad where his guests could travel around and have their drinks and observe what would be the equivalent of the Columbus Zoo in terms of the variety of animals that were there. As a side story I love telling was there were two black rhinoceros that Escobar had in an enclosure there. And I took a couple of pictures of these. At that time, Escobar was on the run, and this was shortly before he was eventually killed. So back in Columbus, I called Jack Hanna and said, well, you know where you might be able to obtain two black African rhinoceros if you're interested. I got a phone call back within a week, and this fellow identified himself as the acquisitions director for the Columbus Zoo. And he said, you know, Jack, I have to tell you that those rhinoceros you saw are not African at all, but those were bred and imported from Texas. You know, on the right slide, this shows the natural habitat for the cotton-top tamarin. Also, further south is Cali, Colombia. Here we were working, our headquarters were in this small town of Zambrano, but Cali is a higher altitude environment and a much cooler environment. We had learned by the grapevine, and this often happens when you're working in an area, that at the zoo in Cali, there was about 30 cotton-top tamarins that had been confiscated from street vendors. You know, exotic in Colombia, the export of exotic animals is, equals to drug cartels in terms of notoriety. So it was illegal for these to be held, and they had been confiscated and were in the zoo at Cali. The zoo director in Cali was a lady named Mara Clara Dominguez. She had attended high school in Minnesota as an exchange student. And she was very happy for us to come to the zoo. They had all the facilities to accommodate primates for we to continue our study on the 30 or so tamarins that were at the Cali Zoo. Next slide, please. So we were able to do endoscopic examination of those cotton-top tamarins at the Cali Zoo and collect biopsies from them. Whoop, what happened? And here are the data. Here are the data from the. Actually, there were 28. There were 30 tamarins, and 2 were pregnant, so we didn't do endoscopy on them. Of the 28 tamarins in the Cali Zoo, notice that 51% of those monkeys had severe ulcerative colitis and moderate colitis, 43%, and never any normal, and just a few with mild colitis in the captive environment in the zoo. Whoop. Here's the comparison of the ulcerative colitis and cotton-top tamarins that we examined during the course of our studies on the animal, comparing Ohio State data with wild living in Colombia. Clearly, high incidence of disease in our captive colony at Ohio State. very little or none in wild living cotton-top tamarins in their Colombian natural environment. In the Cali Zoo, also a large incidence, large proportion of the cotton-top tamarins there with severe or moderate colitis. On the lower right, in the lower right, is data on 6 cotton-top tamarins from the Cali Zoo that the Monterey folks in Zambrano agreed they had a small airplane to fly 6 of them to their back to Zambrano, which was their natural habitat. And we, after a period of, I don't remember how long we waited, about a year, I think, we examined the animals that had been returned to their natural habitat, and all of the severe colitis was gone and some amount of moderate colitis. So essentially what these data show that in the captive environment at Ohio State University or Cali Zoo environment, severe ulcerative colitis, resolution of that colitis when the animals are moved back to their natural habitat, and also none in the wild living population. To the next slide, please. So I mentioned the paper that we had published with this title, Evidence that Colitis is Initiated by Environmental Stress. That's the point I've been making, and sustained by fecal factors in cotton-top tamarins. Now, this prompted us to begin to look at factors within the colon that might be accountable, that might account for so much of the colitis that we've been inclined to study. The next slide, please. This is an electromicrograph of a section from the lumen of a cotton-top tamarin, and notice the variety of different kinds of organisms that are present there. In fact, I'm not sure you can see my pointer, but this one that's attached to the lumen, attached to the surface of the colon, that one turned out to be a relative of a microorganism that causes peptic ulcer disease in the stomach. The next slide, please. The way we approach the question or the hypothesis that fecal factors might be involved in the initiation of the ulcerative colitis or inflammatory bowel disease in the stressed captivity, we did the surgical procedure to prepare what is known as a theory-vela fistula. My colleagues, surgical colleagues at Ohio State, assisted with that. In the center is Donna Caniano, professor of surgery at Children's Hospital. On the left is Khalid Mutabagani from Saudi Arabia, who was a surgical resident. And over here in the center, below the heat lamp, is Michael Stoneroof, who at that time was a graduate of the College of Veterinary Medicine at Ohio State and was staying on for an additional year. And then I don't want to stop screen sharing. No. Here shows a theory-vela loop. The way a theory-vela loop is prepared surgically is that the colon is divided. The guy is divided into 2 parts, and the 2 parts are then then the remaining colon is anastomosed end to end, and the removed loop of colon is then brought to the surface as 2 fistulas. And of course, the advantage of having the 2 fistulas on the surface is we can access the loop with the endoscope. Next one, please. Well, 8 months post-surgery, these are histology sections from the theory-vela loop and perfectly normal mucosa with mucosal cell with mucus secreting cells filled with mucus, no major inflammatory infiltrate, compared with the inline colon in the same animal, again, with the severe colitis. So the take home here is in the theory vela loops, without the fecal stream passing through there, colitis resolved. Next one. So the next question was, what would happen if we were to infuse fecal material in a saline suspension through the theory vela loop? And what we effectively found was that the data, what the data showed us, what with feces infused through the theory vela loop now, there's an increase at 8 months and 15 months post-surgery in a severe colitis now appearing in the theory vela loop, or moderate colitis. And in the main colon do we have to collect data for us to show that just progression of the severe colitis there. So the take home here is that feces infused through the theory vela loop evoked inflammatory disease, severe inflammatory disease, suggesting something in the feces that contributed to the initiation of the colitis and the sustainment of the colitis. Next one, please. So data essentially showed what I've just mentioned, is that fecal suspension infused through the loop increased the severity of the colitis in theory vela loops biopsies and 5 tamarins, as the main colon biopsy just continued showing the colitis as we knew it. The next one, please. Well, when we examined and discussed possibilities for what the stress might be in captivity, what we came to realize was that in the Ohio State University Vivarium, where we kept the animal, the average temperature was 22 degrees centigrade. In the Cali Zoo, average housing temperature was 22 degrees. It was chilly. And what we began asking the question, could it be that part of the environmental stress for the cotton-top tamarins out of their normal tropical environment was some kind of cold temperature stress? So we did some studies, mainly Michael Stonerook in the College of Veterinary Medicine, where the project was to measure oxygen consumption, progressively lower the ambient temperature, and measure CO2 oxygen uptake as an indication of metabolism. What we found was that temperatures below about 30 degrees centigrade, these animals must increase their metabolism in order to maintain a constant body temperature. Because you see what must be happening, and this is what we concluded from this, that the stress of chilly, cold temperatures increasing metabolism was due to these animals evolving near the equator and never involving the mechanisms for adapting to cold temperatures. They don't increase their insulation, for example, by fluffing their fur and other various kinds of behavioral kinds of adaptation to the cold temperatures. And we have evidence for that occurring actually in the natural environment, sometimes at nighttime at higher altitudes. So one of the things we concluded is one possibility is that the stress of the captive environment and in Cali, Colombia, and in the Ohio State University vivarium, that this was a stress factor that may be significant for the initiation of inflammatory bowel disease in this particular model. Next one, please. So to again discuss an hypothesis for the colitis in cotton top tamarins, and perhaps we can discuss a little bit in humans, is that during stress, and we know this occurs, we know quite a lot of the biology of stress. One of the things we know that happens during stress is that there's the compromised mucosal barrier function. In the gastrointestinal tract, the mucosa is essentially a barrier to luminal material, luminal organisms, and various other kind of things found in the lumen, crossing the mucosa into the body per se. That stress, we know very well, and we've studied this at Ohio State, is that we know that stress compromises or opens the mucosal barrier, so that factors in the feces are able to cross the mucosa and into the body. We now know much about how this happens. That is, fecal factors can stimulate some cells, pre-inflammatory cells called mast cells. The mast cells degranulate and release several kinds of substances that then call in the inflammatory cells from circulating from the circulation, call in inflammatory cells into the intestinal wall, and there then is inflammatory bowel disease. Next slide, please. Well, I'll end at this point, and hopefully to entertain any questions any of you out there might have relative to ulcerative colitis, the environment, stress, the microbiota, and anything that crosses your mind.
Can you hear me?
Okay, yes, go ahead.
Yes.
My name's Sam Stout. I'm in anthropology. And it's really interesting because I was trying to, maybe I missed it, but so the temperature as the stress factor, what I'm sure, how did they consider other more behavioral things, like if they're in captivity, does it match their normal population density of animals? Those that induce other kinds of stress because of altering in captivity and in zoos, but it might not be their behavior alterations of their environment, kind of cultural behavior, but primates are pretty social.
Yep. Well, the behavior of the cotton top tamarins is very interesting in itself. In the wild, they form groups of 6, and the group of 6 is never greater than 6. If one of the animals passes on, when it gets killed or picked up by predators, then there's consternation in the column, in the group of tamarins, much agitation, and then they allow another tamarin to join the group. And we observed this. For example, we had several. The company was so accommodating for us with the heavy equipment. We had an enclosure of about 1 acre, fenced enclosure that could not be entered by predators, but the cotton top tamarins were confined there. And what we found, for example, that group of 6, if one of them was, and we had a case where where a predator, a big hawk, picked off one of the tamarins. Now, related to the sensitivity to the cold, the nighttime temperatures fall in Colombia. And the group of tamarins that we were looking at, they lived in the upper strata of the forest. And in that group of 6, one of them was killed by a predator, by a big hawk. Then on the ground, there were another 6 that we had in the enclosure. And in that group of 6, one of the small tamarins, actually one of the small tamarins that was only allowed to join, these groups, we had groups of 20 in the enclosure. A group of 6 would occupy the highest strata of the forest, another one in between, and then there was 6 that were confined to the ground. They only confined, and the only food they could take was what was distributed for them on the ground. The students that were observing this for us were biology students from Bogotá, from the National University. What they observed was when that one small tamarin was killed in the group of 6 that occupied the upper strata, that then what happened was from that group of 6 on the substrate that were on the ground, one of them was allowed to climb the tree and join the group up above. So the conclusion from that was that to maintain body temperature, and one of their mechanisms for adapting is the cuddling, is the grouping together. Often I've seen the cotton-top tamarins in places where they were in captivity, and at night, when you with the light you look to find the group, what you find that all encircling one another. So my point is, and I think this was your question, one of their adaptations, since they don't have the usual mechanisms for adapting to cold temperatures, was to huddle as temperature drops in the nighttime.
Interesting.
What about diet in captivity versus in the wild? I would think that would have a great deal to do with what's going on in the gut.
Absolutely, absolutely, and that was what we surmised as well. And in Colombia, part of the project, we had in captivity, we tried altering diet. We tried altering diet from fruit diet to grain. So I've forgotten exactly what the constituency was of the diet that we tried, but we could never show any effect of diet on the tamarins, either in the Columbus colony or in the tamarins that we maintained in captivity at the research site in Sambano. We thought about this many times with caged tamarins there, but we could never demonstrate any effect of diet on the initiation of inflammation.
Hey, Jack, it's John Bruno from Psychology and Neuroscience.
Hey, John, good to see you, man.
Good to see you again, buddy. Enjoyed your talk. I was wondering whether you had a chance, given the number of animals you had, to look at gender and age in terms of the translation into positive for colitis.
You know, we never did those. We never did those kind of studies. It was just a little bit too much, because we would have needed to do this with the wild population and maintain the animals captured from the wild population and maintain those. So no, we were never able to look at transition from colitis to colon cancer. This is not known, and this would be a very interesting model to pursue that in. But to answer your question, no, John, we never were able to do that.
Vish, do you want to ask your question? Or I could read it. Vish So Ramanan has a question is, was there any follow-up done to test the temperature stressor hypothesis by placing the tamarins in captivity in enclosures that were heated to temperatures closer to what is encountered in the wild?
Could you read that for me, Ardeen? I got part of it.
Okay. Was there any follow-up done to test the temperature stressor hypothesis by placing the tamarins in captivity in enclosures that were heated to temperatures closer to what is encountered in the wild?
Yes, we found Michael Stonerook, when Mike was a student in the College of Veterinary Medicine. We took the equipment to Colombia and did the same temperature metabolism studies on wild-caught tamarins, and we found the same kinds of data, that the progressive fall in ambient temperature at around 32 degrees or so, there must be, in order for the animals to maintain their body temperature, an increase in metabolism. And the ambient temperature metabolic curves were pretty much the same for the wild-caught tamarins caged in their natural environment and those we had at Ohio State.
Okay, and Mark DeBard has a question. What is the significance of your colitis information in tamarins on the occurrence of regional enteritis in humans?
On regional enteritis. Well, in terms of, the questions always come up relative to humans. And I'd point out that it's been known for years that there's a gradient of inflammatory bowel disease and severity of inflammatory bowel disease from Scandinavian countries to the Mediterranean environment. And one can surmise whatever from that, but that's been around for quite a long time. Stress, we're very much interested in stress in the gut and have been at Ohio State. Again, human example, I had a graduate student join me when I left Kansas to Reno, and he brought his wife with him, but his wife fell in love with a cowboy, and Paul, he was divorced. He continued then to have his colitis exacerbated. I was always getting calls to go down to the Washoe Medical Center because his colitis was exacerbated. Dr. Peck tells me that if you have a colitis patient who is a smoker, you never ask him to stop smoking, because the stress of abandoning cigarettes will exacerbate his colitis. The stress story, most of these anecdotal stories, I've always been interested in them, of Edward Keller at the conference in Geneva. There must have been tremendous stress there at that time in the 1940s, just after the end of World War II and the atomic age beginning to stress different people. And the interpretation Owen had of Teller's issue was that he was the stress. He said Jack, it was stress, and in that conference, and we got him back to. And we know this very well in other animal models, and we've done much work of this at OSU on stress in animals such as guinea pigs and mice, things of that nature. That you stress the animal, there's a brain-gut connection that uses, we know many of the neurotransmitters in that connection, sort of thing, that evokes something like an inflammatory response, increases the secretion, leads to the diarrhea and very strong contractile activity in the colon.
I have a question. On the flip side of that, is there any evidence in humans that reducing stress has reduced colitis or other intestinal diseases?
I'm not sure. I was trying to remember what I might I've heard or might know about that. What we do know, there is a brain-gut connection. And we know this mainly anecdotally. For example, Dr. Peck at Palo Alto Clinic, he did some studies once he and I began working together on spinal cord patients at the VA medical center in Long Beach, California. And what the data showed was that if there has been a complete spinal transection in a veteran, for example, that there's nothing in a chart, any of those veterans' charts at the Long Beach VA of ulcerative colitis. And then he always has an anecdote or two about the individual that had ulcerative colitis on a ladder, fell off the ladder, fractured his spinal cord, separated spinal cord, and his colitis was extinguished. So examples like this just become very interesting for someone like basic scientists. We like to think about those sort of things and the relevance. Onother example, Owen did a residency at the Mayo Clinic and was very familiar with the Olmsted County population, especially in terms of medical records. And Owen had a look at all of the compared ulcerative colitis patients at the Mayo Clinic with ulcerative colitis with patients with spinal cord transection. And he tells me the story that he said, we never found any colitis in a spinal cord patient, one with complete spinal cord transection. So this being Owen's way of approaching research, and the reason he was such a great partner is it turned out the guy's wife on the records was a nurse. So he called her up and said, hey, we're interested in the relationship between spinal cord transection with the paraplegia and quadriplegia and the occurrence of ulcerative colitis. And he laughed and he said, you know what she said to me? She said, of course he doesn't have ulcerative colitis because he has a complete. No, she said, of course he does have ulcerative colitis because he doesn't have a complete spinal transection.
Okay, are there any other questions for Jackie?
This is John Bruno again. I'd just like to make a comment. These early studies of Jack's and his colleagues about enteric nervous system and its influence on stress were some of the earliest studies linking the physiology and neurology of the enteric system with the brain. And as you're talking about, Jack, if you interrupted that information from presumably vagal inputs, you lost the stress effect. Is that correct?
Yep. Yeah. Yep. And well, what we're essentially with these anecdotal examples, we're essentially saying if the brain isn't connected to the gut, yeah, individuals don't get inflammatory bowel disease.
Yeah. Well, we have a whole PNI industry now.
So what happens with people when people are being, say, treated pharmacologically for various disorders that affect the brain, those pharmacological things which I know nothing about, that affect the brain? Is there any higher, lower incidence of colitis in these people? Could it be a large sample, I guess?
Well, we know that some of the messenger substances that are involved. For example, if someone is there's an experiment. I have some friends in Barcelona in Spain, and one of the kinds of studies they do on stress and what is released from the brain is that have an individual in a subdued environment in a dark room with low levels of music, and then the individual is asked to periodic with a metronome, every time a metronome dings, is to put their hand into a bucket of warm water, in and out of lukewarm water. And then the assistant changes from lukewarm water to ice cold water. And in the individual can develop diarrhea and several of the stress factors of brain-gut connection. But then you can measure the release of something called corticotropin releasing peptide from the brain, and also from the enteric nervous system, from neurons in the enteric nervous system that make corticotropin releasing factor. So this is definitely one of the factors in the brain-gut pathway that sever the spinal cord and to disconnect those. And we've done so much with this, with the other kinds of other animal models that we stress or lesion the brain or do things of that nature to establish this brain-gut connection.
Howard Warren has a comment that it's perhaps one of the only diseases where smoking has a positive impact on outcome, which is an interesting observation. And then,
well, my point is I've always been told by clinical colleagues that with a smoker, you best not, and if he has a history of inflammatory bowel disease, it's best to not stress him by stopping smoking because of the risk of exacerbating the inflammatory bowel disease.
right, that's it. And then Ole Tuvinen has a question: is there any connection between gut microbes and colitis?
There are connections. I mentioned the organism in this one slide I showed. The one organism that's connected with inflammation and ulcer formation in the gut. It's called a Helicobacter pylori in the stomach, and this was the famous discovery of the association of Helicobacter pylori, the bacterium, with gastric ulcer disease. It's not the same organism, but it's the same group of organisms. I have a friend that in California that showed this. In terms of specific organisms, we really don't know. We do know that for example, we've had a model of parasite infection, and parasitic infection invokes a inflammatory response that we can block by the blockers between that connect brain to gut.
Okay, so it's 5 o'clock. If there's one more question that anybody has, so we can entertain that. And if there's not, then I want to thank Jackie very much, for a very interesting talk, and a talk, that affects many of us, perhaps, and many of our friends. And so I was, very interesting indeed. Thank you very much, Jackie.
Well, you're certainly welcome. I appreciate the opportunity.